Hello and welcome to another week of biology. What you probably have already concluded from this hilarious cartoon is that during this week we're going to explore concepts of heredity. Specifically, we're going to look at those ideas proposed by Gregor Mendel. The first thing I would like to discuss are just some terms to help us understand genetics. These terms will really help you explore ideas and explore concepts. The first term we're going to look at is a gene, or what genes are. Genes are simply those units of heredity. These units of heredity that are passed on from generation to generation. These genes code for specific proteins. We can look at an example of a chromosome, a hypothetical model of a chromosome. You can see that we have a gene, this hypothetical gene A, located on chromosome 1. There's also a hypothetical gene B, C, and D. So you can see on one long strand of DNA that makes up a chromosome, there will be many, many genes. These genes code for proteins that determine specific traits. For instance, gene A right here may code for a protein that controls flower color. Homologous chromosomes are going to be chromosomes that contain the same set of genes. Diploid organisms are those organisms with two sets of every chromosome. There are two of each chromosome. So if we're looking at this organism with chromosome 1, it'll also have a second chromosome 1. That second chromosome 1 will have the same set of genes. It'll still have the A, the B, the C, and the D genes located within that chromosome. These genes code for the same trait, but it might be a different version of that gene or a different version of that trait. Alleles describe those different versions of a gene. So if we think of that A gene represented in chromosome 1, maybe the capital A codes for a gene that determines flower color, and maybe this flower color is red. The lowercase a represented on the second chromosome 1 will still code for a protein that determines flower color, but maybe this one codes for the color green. They're just different versions of that same gene. We call those alleles. As we move on, we'll explore some other additional terms. Another term that will be used over and over again in genetics is dominant and recessive. And what this really means is when both alleles are present, if we go back to that capital A and lowercase a, determining red or green flowers, when both are present, the dominant allele will mask the recessive allele. So in our hypothetical situation, if the capital A red trait is dominant, if the organism has both of those genes, the organism's flowers will be red. The dominant allele masks the recessive. We can see this in one of Mendel's traits that he looked at in pea pod color. When a plant, one of the pea plants, has both genes, in this case the green gene, which is encoded by this capital Y, and the lowercase y, which codes for a yellow colored pea pod, this organism has both genes, but it only comes out as a green pea pod. This tells us that the green gene is dominant over that yellow gene. We progress on this idea and we can look at genotypes and we can look at phenotypes. Genotype is simply the genes that are present within that organism. The phenotype is the physical characteristics of that organism. So for this green or yellow pea pod, we can see that there are 3 possible genotypes and 2 possible phenotypes. There is two dominant alleles for the genotype, which gives a green phenotype. There is one of each of the alleles for genotype that also gives that green phenotype. Or there are the two recessive alleles for the genotype that determine the yellow phenotype. So you can see gene is the genotype and the phenotype is the physical characteristics. One more set of terms to add on to genotype versus phenotype are terms that you probably have heard in the past: homozygous versus heterozygous. Homozygous simply means it has the same set of alleles. Heterozygous means it has two different alleles. And we can see that for homozygous there are two possibilities. It's either those two capital letters or two dominant alleles, or two lowercase letters. Also, we can see that as two recessive alleles. We can differentiate those by saying homozygous dominant or homozygous recessive. There's only one combination where you have both alleles present. We call that heterozygous. And remember, the heterozygous genotype will have the dominant phenotype, or green phenotype if we're looking at these pea pods. When Gregor did these crosses, he noticed that genes separate. Two alleles for a gene, so every parent pea pod plant had two alleles for each gene. And when these gametes are formed, when the sperm and the egg are formed, only one of the two genes is present in the gamete. Basically, only one of the two genes is passed on by each parent. This occurs simply because of meiotic division. If we remember back talking about cell division, we discussed meiosis. During meiosis, those chromosomes separate. This can be illustrated on a simplified version of meiosis, where we have one cell. It has one pair of homologous chromosomes. It has a capital A on one chromosome, and on the other it has the lowercase a. They separate during meiosis, where each of these gametes will have only one of the two possible genes. That means that one half of all of the gametes produced would have the capital A gene, the dominant gene. The other half of the gametes produced would have the recessive gene. The next step would be a fertilization event. So if one of these gametes fertilized another cell, so we had this fertilization event occur, we would get back to that diploid organism with a pair of homologous chromosomes. In this case, the recessive gene in the original gamete fertilized another gamete that also contained the recessive gene, creating an offspring that is homozygous, recessive. What I want to do now is just kind of explore a little bit more into Mendel's crosses. So let's take a look at Mendelian genetics and explore some of Gregor Mendel's ideas. We're going to look at flower color in these pea plants. I'm going to use blue to represent a purple flowered colored plant. So this one is purple. And I will use this black to represent a white colored flower in the pea plant. What Mendel did is he produced these true breeding strains. What true breeding means is if this pea plant is self-fertilized, all of its offspring will be purple in flower color. He would also create true breeding white colored pea plants. So when they are self-fertilized, again, all of the offspring would have white flowers. This was his first step. He called this generation with the true breeding strains the parental generation. The next step, so this is pretty easy to understand. We have different strains of different plants. The next step was the kind of the creative next step. He would cross these two. He would cross the white and he would cross the purple. So now we're going to have this fertilization event between these two pea plants with a different version of those genes. When he crossed these, he didn't see any blending of traits. What he noticed and observed was that all of the offspring were purple. All of the pea plants produced in this cross produced purple flowers. He called this generation the first filial generation, or the F1. His next step was to self-fertilize this F1 generation. And what that produced was 1, 2, 3 purple flowered pea plants for every 1 white flowered pea plant. So that white color reappeared. It wasn't present in the F1, but it was present. It did become present again in this second filial generation, or F2 generation. The next step was to explain how this occurred. So what he determined, what he concluded from this set of experiments, is that each of these parent pea plants contained 2 genes to control the trait for flower color. Based on this F1 generation, he determined that purple was going to be dominant over the white. So we go back to that dominant versus recessive pattern. So if we look at this same idea in terms of Punnett squares, so we can use Punnett squares to illustrate these crosses and illustrate the probabilities of offspring, we can say that we'll use the capital letter P to represent purple and the lowercase letter p to represent the white gene, the white allele. So we always use the same letter. We use the capital for the dominant and the lowercase for the recessive. Being a true breeding purple plant, we know that it has both the dominant alleles. Being a true breeding white pea plant, we know it has both of the lowercase or recessive alleles. So we can use a Punnett square to illustrate this cross. So we create our Punnett square. Parent 1 can go on the top with the 2 capital P's, and the second parent, the white flowered parent, can go on the side with the 2 lowercase p's. These letters on the top represent the genes present in gametes. So these on the top can represent sperm. So remember, during meiosis, these genes segregate into different gametes. The ones on the side can then represent eggs. So the letters on the top and the letters on the side represent gametes. We can bring them down into the boxes and over into the boxes. These boxes represent probable fertilization events. So one probable fertilization event would result in a heterozygous genotype. The phenotype would be purple. And what we notice is that all of these boxes are the same. They're all heterozygous and they're all purple. This is that F1 generation that we see way over here. So we can see through the Punnett square why all of those in the F1 generation were purple. His next step was to self-fertilize these heterozygous purple colored flowers. So we can illustrate that again in a Punnett square. So now we're taking this F1 generation. Remember, these genes will segregate during meiosis. 1/2 of all the gametes will have a capital P. 1/2 of all the gametes will have a lowercase p. Since it's self-fertilization, obviously the other parent will also have a capital P in half of its gametes and a lowercase p in the other half. When we complete this Punnett square, we see one is homozygous dominant, another is heterozygous, a third pea plant here is another heterozygous pea plant, and finally we have one that is homozygous recessive. So we see in terms of phenotype, there are 1, 2, 3 that are purple, and 1 that would be white. Gives us our 3 purple to 1 white ratio that we see here. We show this through the Punnett square. It becomes really a good way to illustrate Mendel's crosses. So hopefully you can start creating Punnett squares and solving Punnett squares through Mendel's crosses. You can do this with the round versus wrinkled seeds. You can do this with all of the different traits, all the 7 different traits that he looked at, and start with that parental generation through the F1 and through the F2. Now the question you may still have is, how does this relate to humans? Now humans, most situations they're going to be more complicated. Mendel looked at very simple examples, but we can still use Gregor Mendel's principles to discuss some human traits. And some of the easiest traits to understand that are controlled by Mendelian principles are those that cause genetic disorders or genetic diseases. These are examples of Mendelian genetics in humans. There are 2 types of genetic disorders when we're talking about dominant versus recessive. There are genetic disorders caused by a dominant allele, and there are genetic disorders caused by a recessive allele. A dominant genetic disorder would mean that the heterozygous individual will carry the gene and have the disorder. A recessive genetic disorder would mean heterozygous individuals will be healthy. An example of a dominant genetic disorder would be Huntington's disease. It's a neurodegenerative disorder caused by a dominant allele. That means if a dominant allele is passed on to an offspring, they would have the disease. A recessive genetic disorder, a good example of that is cystic fibrosis. With cystic fibrosis, the individual to have the disease would have to have 2 recessive alleles. So we can look at this a little bit more through a little bit of a drawing again. So we'll use D's to represent a dominant disorder and R's to represent a recessive disorder. So we have 3 possible genotypes for each. We can have capital D, heterozygous, and we could have homozygous recessive. Same thing for our recessive disorder. We use an R's to represent that. In these cases, who would have the disease? In a dominant disorder, this person would have the disorder. This person would also have the disorder. This person would be the only one that is healthy. In this case, an individual would be healthy. In this case, they're called what is a carrier. They're still healthy, but they carry that recessive gene. This one would be the only individual with the disorder. So we can look at how these disorders can be passed on. The most common way a recessive disorder is passed on is from 2 healthy parents. To pass that on from healthy parents, both of the parents would have to be heterozygous. So this is something like cystic fibrosis. And we can show that if 2 healthy individuals that are both heterozygous reproduce, they're going to have a 25% chance of having an individual with a genetic disorder. With a dominant disorder, it can never be passed on from somebody that does not have the disorder. To pass it on, at least one parent would have to have the disorder. The most common scenario where this is passed on is having one parent with the disorder. This is what generally happens with Huntington's disease. Huntington's disease doesn't manifest usually until after the age of reproduction. So they are able to reproduce, usually with a healthy individual. So in this case, if one parent has Huntington's disease, there is then a 50% chance that they will have a child with Huntington's disease. So we can use Mendel's principles to explore genetic disorders in humans. So we can see Mendel's principles apply to his experiments with pea plants, and they can apply to some situations in humans. Hopefully at this point you can now explore different crosses, looking at genetic disorders, looking at principles of Mendelian genetics. And with that, hopefully you can work through your material and understand the topic.